Hi student, in this video you are going to learn about nucleosome in details and also about the methylation process that happens in the nucleosome as the function to regulate the transcription and translation process of the DNA. So this is the structure of the nucleosome. You may have learned in a cell that the core of nucleosome are made of protein. And here you must know that the name of the protein are histone. It is said here there are 8 histone core protein, which is true. 1, 2, 3, 4 on the first layer, and then on the second layer there will be another 4. So in total there are 8 histone protein as the core of nucleosome. These histone proteins are wrapped around by the DNA strand twice. And then there is another protein histone that is called as H1 protein histone. The function is to bring the DNA that wrapped around to be locked or closer to the core. And then another section here that you must know is called the DNA linker. Based on its name, you may guess that the function is to link one nucleosome to another. So let me draw another nucleosome. So this DNA will be continued, all right? And then it will wrap around another nucleosome, and there will be another H1. So you can see the function of linker, okay? Sorry, the name is linker, DNA linker. The function of linker is to link one nucleosome with another. Now, the 8 protein histone has tails that extend outwards. These tails are called N-terminal tail. Now, the name N-terminal tail comes from the nitrogen that exists in the tail of the terminal tail. Now, what's the function of this tail? During early stage of mitosis and meiosis, chromosomes are very condensed, as you can see from here. Why? Because you know that in mitosis and meiosis, the sister chromatids are about to be separated. So they have to be very short and condensed. Now, this is the function of the N-terminal. As you can see from here, the nucleosome gets very condensed, and then it will become the chromosome that exists in the early stage of mitosis and meiosis. How the nucleosome can be condensed that such way? It is because the N-terminal tail will link up to the adjacent nucleosome. So maybe I can give you an example, like an analogy like Velcro. You know Velcro? Like usually it is used on the shoes or something, that they have fibers like plastic that can, link to one another. So like this. So at the end, this tail will make the nucleosome to be close together, pull them very close together. And this will make the structure of chromosome possible. So what about during interphase? During interphase, cell is very active doing transcription and translation. I hope you still remember this from the SL materials. When we talk about transcription, we talk about protein synthesis. And the cell needs an access to the DNA in the gene. Now, that's why during interphase, this N-terminal tail has to be acetylated. What is the meaning of acetylated or acetylation? Acetyl group will bind to the N-terminal and make the nucleosome loosely packed. You can see from here that the nucleosome is no longer very close to each other. They are moved apart. And by doing this, the DNA that wrap around the nucleosome will be loosed so that the RNA polymerase will have access to transcribe the gene. But not all genes has to be transcribed at the same time. Now, for the genes that are not actively transcribed, they must have their nucleosome to be packed densely, like this one. Who will do that? It's methyl. Remember, for loosely packed, we need acetyl. For densely packed, we need methyl. And the process is called methylation. In methylation, the methyl binds to the N-terminal tail to make the nucleosome densely packed. By doing this, the genes are not accessible to RNA for the RNA polymerase to be transcribed. This mechanism helps to regulate transcription in eukaryotes. Actually, there are two types of methylation. The first one that we have talked about is histone methylation. So the methyl will attach to the N-terminal tail. The second type of methylation is called DNA methylation. The methyl will attach directly to the DNA instead of the histone. We will talk about DNA methylation in the next slide. So now we are talking about DNA methylation. Most of the time, methyl is added to cytosine base of DNA. So this is cytosine. There is C number or carbon number 5 here that is attached to hydrogen. However, if it is methylated, it means that the hydrogen will be replaced by methyl. Now the molecule is called 5-methylcytosine. This process is catalyzed by enzyme and only happens when there is guanine on the 3' end of the cytosine. So let's take a look at this double helix. This is the 3' end. There is cytosine and methyl. Now you can see here, this cytosine has no methyl. Why? Because the base that is located on the side of its 3' end is another cytosine, not guanine. So that's why it cannot have the methyl. However, this cytosine on the other hand, it has methyl. Why? Because the guanine is located on the 3' side of this cytosine. Now this kind of condition, where the guanine located on the 3' side of the C is called CpG site, and if it is methylated, it's called CpG methylation. The process of this methylation is similar, like the effect similar to the histone methylation, which is the gene located in that region will be inhibited from the transcription process. Something interesting about methylation is it is so much influenced by the environment. So for example, we have an identical twins, all right? A pair of identical twins. So are they cute? Okay. So anyway, when they were born or when they were very young, they have similar pattern of methylation because they are identical twins. However, as they are aging, okay, there will be accumulation of differences. This is assumed to be the cause. It is caused by environmental differences. Another data also show the pattern of methylation changes over time during the life of a human. Now, unfortunately, I cannot find a picture or a diagram of it, but you can find in Andrew Ellet page 93. There you can see a graph that shows how CpG methylation changes from a newborn to 103 years old. Now, from that diagram, you can see that newborn has the highest methylation, and then the second one is the 26 years old, and the least will be 103 years old. All right, this can be found in Andrew Ellet page 93. Now, CpG methylation, maximum during birth and decreases as individual gets older. Now, this is a picture that I got from Bio Ninja. So you can see from here, okay, as the twins get older, the differences for methylation increases. So this is supporting the first slide where the twins, as the twins getting older, the differences in their methylation also increases. Right? So the conclusion is methylation pattern, sorry, have a genetic basis but are influenced by the environment. All right. I hope this explanation clear enough. Don't forget to read your ebook as well as the study guide from Andrew Allen. I wish to give special thanks to BioNinja for their wonderful and beautiful picture that I used for this illustration and this video. Thank you so much, and see you later.